Alright, we are back with proteins and nucleic acids. Proteins, just like carbohydrates and lipids, are composed of carbon, hydrogen, and oxygen. But unlike carbohydrates and lipids, they also include nitrogen, and most of them have sulfur. Sulfur is in a couple of amino acids on the side chain. So if you don't remember, the monomer of proteins are amino acids. They're going to be abbreviated AA. All of them have a CH bond in the middle. They have an amine group here, which is positively charged. And they have a carboxylic acid group, okay, COOH. They also have this side chain, R, okay. There's no element R, but this R represents something that can change, okay. So this R group is the only thing that's changing per amino acid. They are joined together using a peptide bond, okay. So when you join more than one amino acid, they're joined together using a peptide bond. Remember that over here you would have a nitrogen, a carbon, and a carbon. This bond right here is a peptide bond, okay. A peptide bond is formed between the hydrogen of an amine group of one amino acid and the hydroxide of another carboxyl group. Okay, so we've got one amino acid here, over here, and then when we join them together, we've got a peptide bond. This bond right here is a peptide bond. Very, very important, okay? Which means this right here is called a dipeptide. Another name for a protein is a polypeptide. Important. Poly meaning many. Again, when we join amino acids together, we are creating water, which makes this a condensation reaction. Okay? Condensation reaction because water is formed just like the other biomolecules. When you join smaller monomers together, you create water in a condensation reaction. Proteins are found in many different parts of the animal cell and also in your body. Some common examples are protein channels in the cell membrane. Okay, things that cannot fit through the phospholipid will take this protein channel because there's a larger opening. And we'll talk more about that when we discuss the cell membrane. There are also proteins embedded throughout the cell membrane. Some of them serve for recognition and things like that. Antibodies are made of proteins. These are very, very important in the immune system. If you have questions about that, you're welcome to ask me, but we cannot go into that right now. The most important thing that we are going to study with proteins is something called an enzyme. Okay? And an enzyme is simply something that will speed up a chemical reaction. It's a protein that changes the rate of a chemical reaction. It always ends in ASE. Okay? It's typically called a catalyst. And again, a catalyst is something that catalyzes or speeds up a reaction. A catalyst increases the rate of a chemical reaction by decreasing the activation energy. And I will show you a graph on this in a second. Okay, right here. We've got a graph right here. This graph looks complicated, but let's break it down. Okay? If I just look at the chemical reaction, so any chemical reaction, we're converting two things to two other things. Okay? This might be acid plus base equals salt plus water, for example. Okay? But there's a chemical reaction happening, and there is an energy involved to get that reaction going. It's called the activation energy. Okay? So activation energy is the amount of energy it takes for a reaction to occur. If we follow this normal path right here, okay, going up, this is looking at the progress of the reaction. So here we have our reactants. we have our reactants. We still just have the reactants. As soon as we get to the activation energy, then we have enough energy to start converting things to products. So that's showing down here. Once we get back down to this line, then all of our products are formed and we have no reactants left over. If we're talking about our acid plus base and salt plus water, then we'd have all acid and base over here, all acid and base, all acid and base. Over here we'd have a mixture of acid, base, salt, and water. More salt and water, more salt and water, and then down here we have all salt and water. Okay? So over here we have reactants, and over here we have products. The only thing an enzyme does is speed up the chemical reaction. It lowers the energy required. So as you can see from this graph, we hit that activation energy a lot quicker. Okay? Because it does not take enough energy, it does not take as much energy for this reaction to get started, it therefore does not take as much time. There are a ton of chemical reactions that would take way too long if we didn't have enzymes. Okay? So this is called a catalyzed reaction when it uses an enzyme that's functioning as a catalyst. It can only work under certain environmental conditions, and this is what our lab is on. Okay, so we'll explain later on. Enzymes work like a lock and key, so you need to remember the lock and key model. A certain enzyme has a certain shape, and it will only work with a certain substrate. Okay, the substrate is something that is locking into the enzyme. So here's your enzyme, here's your substrate. For example, this could be lactase. Notice that it has ASE on the end. That means it's an enzyme. The sugar that's involved here is lactose. Notice it has OSE on the end. Okay? And this is just an example. There are many thousands of enzymes and many thousands of substrates. Keep in mind that only one substrate will fit into each enzyme. Okay? So lactase will only break apart lactose. It's not going to touch other types of sugar. Okay? This part right here is called the active site. There are certain sequences of amino acids, remembering this is a protein, that make up the active site. These are the important ones for catalysis. Okay, so reaction of a substrate into an enzyme. Right here, these amino acids are the ones that are going to determine the function of the protein. So keep that in mind when we get later on into genetics. We'll talk about mutations. If you mess up these amino acids, the enzyme's not going to work. Okay? So a substrate fits into an enzyme like a lock fits into a key. Only one will work. Okay? Only lactose, for example, will fit into lactase. When that happens, the chemical reaction occurs and it will break apart the substrate into its products. Okay, so we're converting the reactant to products. Remember that lactose is a disaccharide. So when we break it apart, we'll break it into two monosaccharides. There's an enzyme in your body that does that called lactase. Alright, proteins are used for building, replacing, and maintaining tissue. They're also used for movement. They are also used for gene regulation. Okay, so we'll talk about that again more with genetics. They are used for chemical reception. A good example is insulin. And if you want to go look up how diabetes works before we actually talk about it in class, that's probably a good idea. It's also used for chemical storage. Okay. For example, in your body, there's a really important enzyme. The name of that enzyme is hemoglobin. It carries oxygen in your blood. Okay, carries oxygen. It's very, very important that hemoglobin is structured the way it is so that you can carry enough oxygen to your cells for cell respiration. And of course you know that cell respiration produces energy, and that's why it's so important to have that oxygen. They're also important in the immune system. Again, we talked about antibodies and things like that. All right, nucleic acids. We are going to completely learn about nucleic acids when we get into genetics, but for the cells unit, you just need to know the macromolecular structure. Okay, again, we've got carbon, hydrogen, and oxygen, just like all the others. We also have nitrogen, and we also have a phosphorus. Okay, phosphorus is important in every nucleic acid, and if you remember, the monomer of a nucleic acid is a nucleotide. It's made out of 3 things. Okay. Sugar here, 5-carbon sugar. In DNA, remember, it is called deoxyribose. Okay? And in RNA, it's just called ribose. We have a phosphate group. A phosphate is PO4, okay, it has phosphorus and oxygen. You might recognize it, okay. And then the base, you might recognize these also. The bases are A, T, C, G, and U. So adenine, thymine, cytosine, guanine, uracil. Okay, and we'll get in more about that later. All right, this is the molecular picture of a nucleic acid. They are stacked on each other, okay? And again, when you join 2 nucleotides, you are creating water in a condensation reaction, okay? So here is your phosphate, here's your sugar, here is your base, okay? This one is adenine. It's got a double ring. All right, nucleic acids. There are 2 examples of nucleic acids. There's only 2. There's DNA, which is deoxyribonucleic acid. It's wound up into a double helix. Again, we'll get more into the structure. I like this picture. This is a chromosome. Okay, we're unwinding, getting smaller. You can start seeing the helix. Okay, as we get down to a molecular level, we know that this backbone right here is made of sugar and phosphate, sugar, phosphate, sugar, phosphate. And these bases, okay, are in the middle, right? So I know you paired these letters before, but now you hopefully will start to understand what they actually mean. So this right here, this backbone, this is the sugar phosphate backbone. So we've got deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate. And then in the middle, we've got these bases. Okay, so guanine pairs with cytosine, adenine pairs with thymine. And again, we'll get more into that later. RNA has the same basic shape, okay? RNA is single-stranded, so there's not this double helix typically. And RNA has the sugar ribose instead of deoxyribose, and that's why it's named that. Nucleic acids are used to carry genetic information. So as we get further into genetics, we will look at these processes. They are called transcription and translation. What you need to know now is that nucleic acids carry the code to determine the sequence of amino acids in proteins. Okay? So DNA carries the instructions for how to set proteins up. Remember, there's only 20 amino acids, and that structure determines what the protein is going to do, because structure determines function. This controls all of body processes, okay? But keep in mind that every single aspect of the way you look and arguably the way that you act in some cases is controlled by your genetic information, which comes straight from DNA. It's also used to help protein synthesis, okay, of course. So we'll talk about that more in genetics.